Thanks for the invite to speak today. I'm David McClymont. I'm the head of automation at the London DNA Foundry, and we are located at Imperial College London, and we're also co-located with Simby City, which is the Innovation Knowledge Centre for Synthetic Biology in the UK that some of you might be familiar with. And the reason I mention that is because it's a really nice place to be in that, you know, we can take some great academic research in the lab, but also we partner with companies to make sure and accelerate the industrialization of these processes. So it's a really great mix of two great things, and obviously the automation platforms we've built and designed there has been developed to meet those needs. So here's a small picture of our Foundry, and I think what we're trying to do is have multiple work cells. So by having different systems that are focused around, you know, one generalized activity with, you know, potential for overlapping functions, what I'm doing when I create a Foundry like this is I'm looking for multiple solution paths, because I find it's very difficult to predict exactly what equipment or technologies you'll need, and also it's difficult to code and manage that entire system unless you've got some sort of separation in there. So, you know, the way we've designed that is we've got the lab site access, you know, acoustic dispensing with all the kind of integrated instruments you would expect, centrifuge and plate sealer, but also two different tip-based liquid handling work cells from SciBio, you know, working with cultures and things like magnetic bead purification. So, you know, what I've shown here is, you know, the automation technology, but, you know, we've also got a lot of metrology technology like mass spec, but, you know, these boxes tend not to photograph as well as these things. But, and kind of bringing the whole thing together, the philosophy that we wanted behind that was everything that we do starts with design of experiments. So we've bought the software Jump from SAS, which allows us to do DOE and statistical modeling, and that feeds into how we design the automation. So here's just an example of one of the automated yeast build process we have. Like I said, it starts off with the design of experiments, and, you know, we use the SBOL visual to sketch out what we're trying to do. But what Jump does, which I think is really great, is it allows biologists and, you know, who have been a little bit, you know, not up to speed with, you know, robust statistical methods to really start doing DOE from the very beginning. And what I realized is that DOE could be quite easily made into an automated build procedure, in this case for the yeast YTK. So the software platform I've built is essentially, it's not quite a LIMS, it's not quite you know, a driver, but what it does is it takes the context of the experiment as designed by the DOE and actually turns it into something that's automatically made on the work cell system. So for example, you know, if you want to make 1,000 designed organisms, we're talking around about 15,000 individual liquid transfers in and out of about 14 different assays, which actually isn't a lot if you come from high throughput screening like me, but as these signals multiplex and come together in and out of different size plates, then, you know, that software platform really had to be developed to do that. So, you know, working on yeast a lot, E. coli with a technology called BASIC, which was developed at Imperial, you know, a good example of that tech transfer, and also cell-free with the same principles. So, you know, as these things are built, you know, we automatically do that on the work cells. You know, then we move into the high throughput screening. So, you know, characterization, growth curves, and also targeted mass spectrometry. But also we're collecting and developing the workflows for the metabolome and the proteome. And, you know, my worry is that, you know, as we collect all that data, that, you know, what are we doing with it? And because it's been designed with the DOE in mind, you know, our hope is that that's going to be informative as we go on, and that's why we've built up that pipeline. And, you know, as it started with the DOE and as the build was initiated by that, you know, statistical modeling is now very simple, because, you know, response surfaces and, you know, modeling that entire, let's say, operon that we're interested in, I think is much more informative. And I think it allows our scientists a really, you know, easy to use starting point to start to really think about these pathways and data in a more robust manner. So, you know, how do you build out these systems. So, I think, if something's working in a lab or someone's got a nice experiment, it's not just a straightforward process just to say, well, let's automate that. Let's have a robot do that. And some of our, ideas were published last month in Nature Biotechnology. Automation isn't anthropomorphic. There is a process that I've developed over the years which I call, similar to the Agile manifesto within software development, is, how do you break these things down into manageable chunks and build out these systems in a scalable way? So the way that I do that is, breaking things down into small work packages. Look at the entire process that's been developed and break that down into little chunks. And when you break down into the chunks, you can then have appropriate QC that you can say, well, this part of the process can now be introduced into the workflow. But also with that QC, want to find out what the range of that is. Where do things break down? Where do your assumptions stop? And that can be just as important as meeting a general QC step. But I think when you do that as well, when you're trying to build these systems out, I think really what I'm also looking for is shortcuts. You might find that a difficult, trying to cut corners. But really, in order to build out these systems in a scalable way, you just have to take them apart. Sometimes you have to go back to the original way that things were done and find a little trick that allows that to happen, or potentially, you might have to say we need new technology there. So this agile development with the iterative cycle, has proven quite successful for us. So we opened in April 2016. And, I'm not going to go through all of these, but, the timeline shows some of the systems that we've brought online. And, it's an important distinction to say, what we're trying to do here is we're trying to build scalable, plate-based, automated workflows. It's not, a narrow research interest. Everything we do, has to become a scalable process so that we can turn the handle and, use it time and time again. And I think that key part comes from where we are at Symbicity. Between the industrial and academic partners, we can focus on building out these platforms. A number of these things, E. coli, yeast, and cell-free, developed these principles that the handle can be turned on those. So I've been asked to, talk about challenges, and in actual fact, I think the biggest challenge for automation is culture. The technology for doing these things has been in place for quite a number of years. The pharmaceutical industry has essentially beta tested the whole thing, and while coming together and by hand making a quilt might be quite a fun thing to do on the weekend, I think, coming together and by hand putting together bits of DNA to make a genome sounds, less fun to me. And I think that, at this time, we should probably commit to, really quick tech transfer of existing or new automation technologies, with dedicated assay development teams in Foundries in order to do this in an industrialized, scalable way. So that's what I think has, been the main challenge over the years. And this is our team in London outside the Albert Hall. So that's my talk, and if anyone has any questions, I'll be happy to answer them. Thank you. So we can do about 1,000 organisms a day. So that's running through around about 15,000 transfers and about 14 plates. So the other advantage of having the different work cells is that we can batch process things. We don't need to go to the end of execution. So we can get a lot of scalability around saying, well, this process gets done to this point, and then these things are banked, and then the next day we can do the next section. When you're pulling out enzymes or pulling out different bits of material, I think a lot of the scalability comes from thinking about the process as well as and how that fits into the equipment you have.